All right, everybody. Well, it is 9 a.m. and let's get this party started. So I just want to start off by saying, please join in. As you figure out about this meeting, as you figure out the Google Meets. So the way that we're going to run biology now is off of the new master schedule. So once a week, I will be doing a extended biology lesson. I'll be doing it live. I'm live right now. You could pop in if you wanted to, say hi. You could participate. You can ask questions, follow along with the readings, whatever you want to do. As we move on, we're going to have like 4 or 5 more weeks of this. I'm going to really encourage you guys to join up live. It'll be fun to chat. It'll make the videos more entertaining, and I'm sure you'll get something out of it. Guarantee you that. So let's see if I can get my screen to present. Present my entire screen. Okay, so you should still be able to see me. And what I've switched over to is our weekly activity packet. So last week and the week prior, what did we study? We covered, we've covered carbohydrates, which you can see here. And we've covered lipids. You know that I used to talk about Amoeba Sisters during class. I still think they're a really great resource. So I found a couple of really nice educational gifts that actually fit perfectly along with our study of nutritional macromolecules. As you can see, let's wait for this animation to run again. Here's our little glucose molecules that we studied in diabetes. We talked about monosaccharides, we talked about disaccharides and polysaccharides, which are just longer chains of glucose molecules, right? We've talked about lipids, where we have these glycerol molecules and these fatty acids, sometimes called triglycerides. Those are the types of fats, too, that you'll see saturated, unsaturated, trans fats. Those all exist in lipid form. Today, what we're going to cover is all the way to the right. We're going to look at proteins. Excuse me. I know a lot of what we think of proteins are just like, eating meat, eating beans, drinking milk, protein shakes, bodybuilding, stuff like that. That's completely true. Your muscles are made of primarily proteins, and that's like what allows you to like have muscular movement is actin and myosin filaments, which are made of proteins themselves. But the fascinating thing about proteins are they basically do every action inside the human body. Anything that's, sorry, something keeps popping up on my screen and distracting me. Anything that's performing an action, anything that is an enzyme, performing any sort of reaction, moving anything, those are all proteins. So what I want to do first is check out an article. If you click, all right, so let's go back to, I want you to keep looking at my screen, so we'll go to Google Classroom. Sorry if you can hear the construction next door. I know that's no good. So we'll go to any one of your classes here, and you'll notice that here is my announcement for the day, just saying that I'll be going live soon. I've got the classroom codes, both nickname and official code. So if one doesn't work, try the other. And then up here, what you'll see is your weekly assignment. So instead of posting small chunks every single day for you to do, basically what we're doing is we're trying to condense everything down into one nice live meeting. All right. So that's where I'm getting this page. All right. This will be due next Wednesday. I'm going to give you a week to do this. So each class that we have on Wednesday, the material will be due on the following Wednesday. So let's check it out. This week we're looking at proteins. I figured it'd be good to start with a text article because we haven't done any text in a very long time. Let's see if anybody else is here. All right. My original plan was to do popcorn, but since it's just me, you guys are going to be blessed with my voice in this article. So the article reads, Introduction to Proteins and Amino Acids. What are they? Introduction. We tend to think of protein as a mass noun, a homogeneous substance. Homogeneous means something that is very uniform, by the way. So your milk in your fridge is homogenized. That means there's no fat layer on the top because it's uniform. More natural milk that hasn't been shaken up or homogenized, that's the sort of stuff you find with the fat cap on top. So milk with the cream on top, not really a fan, to be honest with you. But that is a non-homogenized milk. So we tend to think of protein as a mass noun, a homogeneous substance, something that your diet should contain in certain proportion. But if you ever work in a molecular biology lab, let's say for a summer internship, protein may start to look very different to you. How so? Well, you may see firsthand that protein isn't just a single substance. Instead, there are a lot of different proteins in an organism, or even in a single cell. They come in every size, shape, and type you can imagine. And each one has a unique and specific job. Some are structural parts, giving cells their shape or helping them move. Others act as signals, drifting between cells like messages in a bottle. Still, others are metabolic enzymes. These are my favorite personally, putting together or snapping apart biomolecules needed in a cell. And the odds are, one of these unique molecular players will become yours for the duration of your research. What they're referring to, that research, they're talking about hypothetically, if you're in a summer internship program, there is going to be no large research project relating to one of those macromolecules in my class anyhow. Proteins are among the most abundant organic molecules in living systems and are way more diverse in structure and function than other classes of macromolecules. A single cell can contain thousands of proteins, each with a unique function. Although their structures, like their functions, vary greatly, all proteins are made up of one or more chains of amino acids. In this article, we will look into more detail at the building blocks, structures, and roles of proteins. Types and functions of proteins. Proteins can play a wide array of roles in a cell or an organism. Here, we'll touch on a few examples of common protein types that might be familiar to you and that are important in the biology of many organisms, including us. Enzymes. Enzymes are awesome. They're very fascinating in my opinion. Enzymes act as catalysts in biochemical reactions, meaning they speed these reactions up. Each enzyme recognizes one or more substrates. Substrates are the different molecules that serve as starting material for the reaction it catalyzes. Different enzymes participate in different types of reactions and may break down, link up, or rearrange their substrates. One example of an enzyme found in your body is salivary amylase, which breaks amylose, a kind of starch, down into smaller sugars. The amylose doesn't taste very sweet, but the smaller sugars do. This is why starchy foods often taste sweeter if you chew them longer. You're giving salivary amylase time to get to work. So this is actually really fascinating. I'm breaking off the reading for a second. We were talking about starches last week, 2 weeks ago. Starches are just really long chains of glucose, right? I don't think potatoes taste sweet. And I hope you don't either. Unless, of course, it's a sweet potato. Oh, it's too early for these jokes. I am so sorry for all of you who are watching this. The reason potatoes taste rather bland is because their glucose is connected in very long chains. When glucose is in very long chains, it's considered a starch. It doesn't taste sweet, right? Potatoes don't taste sweet. Chips normally don't taste sweet. Pasta is normally not sweet. But if you chew it for a very long time, there are enzymes in your mouth, in your saliva, in your spit. Before your food even gets swallowed, it's already being digested in your mouth. So those long chains of glucose stored as starch are starting to break up into smaller, more sweeter and tastier versions of that same molecule in smaller pieces. Another form of proteins, hormones. You may have heard of these. Hormones are long-distance chemicals released by endocrine cells, like the cells of your pituitary gland. They control specific physiological processes, such as growth, development, metabolism, and reproduction. While some hormones are steroid based, you can check out the article on lipids, others are proteins. These protein based hormones are commonly called peptide hormones. For example, insulin is an important peptide hormone that helps regulate blood glucose levels. When blood glucose rises, for instance, you eat a meal, specialized cells in the pancreas release insulin. The insulin binds to cells in the liver and other parts of the body, causing them to take up glucose. This process helps return the blood sugar to its normal resting level. That's interesting. Personally, I never thought about insulin on that level. It completely makes sense that it is a protein. All right, check these out. Just a couple different types of proteins and their functions here. So you have digestive enzymes. Amylase is breaking down starch in your mouth. Lipase and pepsin, I believe, work in your body, not in your mouth. Hemoglobin, we all know this one. Hemoglobin transports oxygen through our blood. Actin is in your muscles. Keratin, I can't remember if keratin is hair or if keratin is skin. Hair. Keratin is in your hair. Tubulin. Gotta look this up. Okay, so this one right here, tubulin, this helps make your cellular cytoskeleton. Here, I need to get a piece of paper. This is a really bad picture. Even though our animal cells do not have cell walls, there are still structures within their cellular membrane, and these structures are sometimes built by, well, they are supported by tubulin, and it's basically like an internal skeleton for the cell, if I want to put that loosely. Let's see what else we have here. So hormone signaling, we have insulin and glucagon. We've studied those in Unit 1 with the relationship to homeostasis and diabetes and blood sugar management. Defense mechanisms. So you have antibodies, right? Antibodies protect the human body from foreign pathogens. Contraction, myosin. So actin is part of your muscles. Myosin is also part of your muscles. Okay. Carry out muscle contraction. And then the legume storage proteins. So that's basically saying like beans. Beans are high in protein. You have energy stored as proteins. And then the egg white. So the white of your egg, when you crack an egg and, it's on the pan, what turns white is called albumin. That is protein that the chick would normally consume during development, right? Because obviously an egg is an unborn chicken and ours aren't fertilized, the ones that we eat. So in the case of fertilization and that egg matured or that chick matured inside the egg, it would be consuming the albumin. Oh, wow, quite a bit to go in this article here. I'll speed up. So, proteins come in many different shapes and sizes. Some are globular, roughly spherical in shape, whereas others form long, thin fibers. For example, the hemoglobin protein that carries oxygen in the blood is a globular protein, while collagen, found in our skin, is a fibrous protein. A protein's shape is critical to its function, and, as we will see in the next article, Many different types of chemical bonds may be important in maintaining its shape. Changes in temperature, pH, as well as the presence of certain chemicals may disrupt a protein shape and cause it to lose functionality, a process known as denaturation. So when a protein denatures, it's basically dying. It's losing its function, it's failing to do its purpose. Amino acids, the building blocks of proteins. So amino acids are the monomers that make up proteins. Specifically, a protein is made up of one or more linear chains of amino acids, each of which is called a polypeptide. We will see where its name comes from a little further down the page. There are 20 different types of amino acids commonly found in proteins. That's also true for us humans. We have 20 amino acids that make up the entirety of our protein array. Amino acids share a base structure, which consists of a central carbon atom, also known as the alpha carbon, bonded to an amino group, which is consisting of a nitrogen and two hydrogens, a carboxyl group, which is abbreviated as COOH, one carbon, two oxygen, and one hydrogen, and a hydrogen atom. You can see this here. We have our amino group, we have our carboxyl group, our hydrogen atom, and then our fourth charge on our central carbon atom is occupied by a generalized R group. Although the generalized amino acid shown above is shown with its amino and carboxyl groups neutral for simplicity, this is not actually the state in which an amino acid would typically be found. At a physiological pH, which is the pH that we exist at, 7.2 to 7.4, the amino group is typically protonated and bears a positive charge. That means that there is additional protons on the amino group, and this side of the molecule actually has a positive charge. Just like a magnet has a positive and a negative side, that can happen on molecules as well. The carboxyl group is typically deprotonated, which means it's lost some of its protons, and it typically bears a negative charge. Every amino acid also has another atom or group of atoms bonded to the central atom, known as the R group, which determines the identity of the amino acid. For instance, if the R group is a hydrogen atom, then the amino acid is glycine, whereas if this R group is a methyl CH3 group, the amino acid is alanine. Whatever, so I made a little mistake earlier. Whatever the term the 20 common amino acids are shown in the chart below with their R groups highlighted in blue. So if we look back to the general, we see our amino group NH2, right? And then we see our carboxyl group COOH. This is our carboxyl group COOH. This is our amino group. And this is our chain. Arginine, histidine, lysine. Here, let's zoom in. You can see. We've got our amino group, our carboxyl group, and then our this is what makes this histidine. This R group is what makes this lysine. Oops. Yeah, let's So one other thing that I want to 21 amino 21. Hmm. Let me just double check something on my other computer really quick. Okay, so humans can produce 20 amino acids, but I'm seeing this chart is very common, the one that we're looking at right now. So I'm trying to figure out which one is the one that we can't, or the one that's not on this, one that's not supposed to be on this chart. All right, let's move forward. So it says the proper amino acid's chemical behavior that is considered acidic, basic, polar, or nonpolar. For example, amino acids such as valine and leucine are nonpolar and hydrophobic, which means they are not charged and they do not like water. Amino acids like serine and glutamine have hydrophilic side chains that are positively charged. Some amino acids, such as lysine and arginine, have side chains that are positively charged, logical and are considered basic amino acids. Histidine is also sometimes put in this group too, although it is mostly deprotonated at a physiological pH, which means it's lost its protons, which means it's not charged anymore. On the other hand, are negatively charged at a physiological pH and are considered basic. So if you scroll up here, you can see they've organized the 21 amino acids into this chart. So we can see what their chemical and reactive properties are. A few other amino acids have R groups with special properties, and these will provide important when we look at protein structure. So proline has an R group that is linked back to its own amino group, forming an exception to the since it no longer has the standard NH group. If that a ring structure looks a little awkward, you're right. Often proline bends causing proline often. Let's look at pro. See, this is particularly odd because you see here, the amino group is still exposed on all of these other amino acids. So yes, they have rings, but they still have their independent amino groups. Proline seems to be very fascinating because we're actually not going to watch the video on protein structure because I feel like where we are right now with this study, that's all we need. We just need pretty much a surface level understanding of protein folding and protein structure. I'm going to skip that. Is made up of in a specific order. A polypeptide is kind of like a long word that is spelled out in amino acid letters. The chemical properties and order of the amino acids are key in determining the structure and function of the polypeptide and the protein it's part of. But how are amino acids actually linked together in chains? the amino acids of a polypeptide are attached to the neighbors by covalent bonds known as peptide bonds. Each bond forms in a dehydration synthesis, also known as condensation reaction. During protein synthesis, the carboxyl group, the COOH group, of the amino acid at the end of the growing polypeptide chain reacts with the amino group of an incoming amino acid, releasing a molecule of water, just like we saw with that was starches, right? We saw dehydration synthesis in the formation of long glycogen chains. During protein synthesis, the carboxyl group of the amino acid, and I already read that part, my apologies. The resulting bond between amino acids is a peptide bond. So this is what you see here. We have an amino acid and an amino acid. They're being joined together to form a chain of amino acids, which is known as a protein, also known as a polypeptide. We were actually doing this. We did this during our diabetes unit. Remember when we actually took a DNA sequence, we transferred it to mRNA, and then we transferred it to tRNA, and then we broke it down into codon sequences, which were little chunks of 3 nucleotides, and then depending on whatever nucleotides you had, that would bring in the correct transfer RNA holding the correct amino acid. You guys have actually done this before. You're experts. Because of the structure of amino acids, a polypeptide chain has directionality, meaning that it has 2 ends that are chemically distinct from one another. At one end, the polypeptide has a free amino group. At this end, it is called the amino terminus, or N-terminus end. The other end, which has a free carboxyl group, is called the carboxyl terminus, or C-terminus. The N-terminus is on the left, and the C-terminus is on the right, for the very short polypeptide shown above. So pause and think. Which side would this polypeptide be able to add to? So would we be able to add to the N side, or would we only be able to add to the C side, based on what we just read earlier? I'm just checking to see if we've got anybody. All right. On the other end. The functional protein between amino activity page here. You all should have your own copy. Obviously, pick English or Spanish, whichever one you're more comfortable with. So we knocked out this article, right? And let's see what else we have today. So just as a reminder, what I've done is I've taken our week-long activities and I've condensed it into an hour and 20, hour and 30-minute lesson for everybody. So let's check out video number 1. Introduction to amino acids. As a reminder, amino acids are the smallest available molecule that can be used or can be considered a building block for a protein. Just like the smallest available building block for glycogen was a glucose molecule. Let me close this article. Let's read through the 6 questions that I've set up for the first video. Question number 1. Proteins are made up of chains of blank. I've lost connection. How many amino acids are there in most biological systems, including humans? 10, 23, 20, or 46? What elements, question number 3, What elements compose an amino group? Nitrogen and hydrogen, oxygen and carbon, or carbon and hydrogen?